Hi, welcome to lesson number 3 on our mitosis lessons online. And today we're going to talk about cell cycle clock and cancers and how we're going to use calculating the different phases of mitosis in our second experiment for Tuesday. Okay, well, let's begin. So here's the cell cycle clock. The cell cycle clock is a series of chemicals that give signals to the cell's nucleus to begin cell division. So let's start. In the beginning, right after the end of telophase and cytokinesis, the cycle begins in what we call G1 or the gap. Then there's an S phase. During the S phase, that's when the DNA is replicated. And then it goes into gap 2, which then signals the cell to undergo mitosis. At the end of this, you end up with two identical cells, and the cycle continues. There are two types of chemicals that play out after cell cycle synthesis in gap 2. The first is a tumor suppressing gene, which is like A brakes, and a proto-oncogene, which is like the gas. So a proto-oncogene, if there's a mutation in that, causes the cell cycle gaps to shorten and mitosis to happen more often. The tumor suppressors like the brakes, and if there's a mutation in the tumor suppressor genes, then the cell then proceeds through the gap 1 and gap 2 without brakes, and again, uncontrolled cell division. So when we're looking at the cell cycle clock, we're really looking at how these two chemicals, proto-oncogenes and tumor suppressor genes, can play out in what we call cancers. So what you have here is a comparison of male and female cancer rates. And if you'll notice, there's the prostate and the breast cancers are near the top, and then all the way down to the pancreas and urinary bladder cancers for men and women. So cancers are pretty scary stuff, and that's one of the reasons why I became a teacher, was so that I could teach one of my students biology, and then eventually they would find a cure for cancer. So good luck to all of you. Hopefully you'll find that out. So let me walk you through a brief explanation of what happens. Okay, so a healthy cell goes through some kind of mutation, and the DNA, either from exposure to chemicals or drugs or smoking, and there's a mutation in which the DNA is mutated, and then that causes, and if that mutation occurs in the proto-oncogene or in the tumor suppressor gene or in both, you then begin to have these sets of uncontrolled cell growth. And because that mutation is replicated during mitosis, you'll have two cells that have the same mistake, and then four cells that have the mistake. And then eventually what happens is you have this now groups of cells that are growing uncontrolled, but in a localized situation. We say that's benign or a bad cancer, but not too terrible. But if left untreated and it continues to grow, then those cells begin to move towards the circulatory system. During this time, the tumor begins to call for capillaries for blood, and as one of those cells enters into the capillaries, then we say it's metastasized or really bad. Now those cells that have no controls continue to replicate and then produce more cells in other places. And that's when the cancers have spread. So cancer is basically a mutation in the DNA that causes the cell cycle clock to speed up its cell divisions without any kind of control or break. So you're making identical cells with the same mutation, and you're making it at a faster rate than the normal cells around it. And we begin to call that cancers. All right, so tomorrow, or on Tuesday, you'll be looking under the microscope, and we're going to try and figure out the cell cycle clock. How long are the cells in interphase, prophase, metaphase, anaphase, and telophase? So you're going to look under the microscope and you're going to try and find this image, usually on the 10 or the 100. You can then move to the 400, that's the highest power, and you should begin to see these cells. When you see these cells, this is called a field of view, so what you see in the microscope on the highest power. And then you're going to count the number of cells that you see that are in each of these phases. So you'll see them, here's anaphase, telophase, metaphase, metaphase. Here's another metaphase. This one right here looks like prophase because the cell nuclear membrane's disappearing. Here it looks like you have telophase and cytokinesis because they've just finished. Here's another anaphase, and when you see this blue dot in the cells, that's interphase. And if you don't see the blue dot, or if you don't see a distinct nucleus, then it's prophase. So you're going to count up in 3 different views how many cells do you see that are in each of those 5 phases. All right, hopefully that helps. We'll see you very soon.